Good morning, everyone. First, I want to say thank you for being here. And I just want to also, in addition, say thank you to Lab Conscious, to New England Biolab, and the Lab Central for organizing this. I usually been going around doing talks about environmental impact and such, but I just want to point out you are my favorite audience because I know we all have one thing in common. You guys are all here because you care about the environment, you want to reduce down the waste that your labs are producing. So we're also going to have one more thing in common, and that's cost savings. We all like to save money, and that's something else that Grenova also going to be able to provide that to the industry. Grenova stands for Green Innovation, and how many of you guys have heard Grenova before? Raise your hand. All right, two. Yeah. So we are fairly a young company, about 5 years, and I worked in the industry in the lab automations for over about 15 years. And what I noticed that as our industry in life sciences, doesn't matter your clinical lab, molecular lab, agrosciences, we're introducing automations into our labs, and our throughput is rapidly increasing. But at the same time, the amount of plastic waste that we're producing, it's skyrocketing. And it seems that was something that everyone neglecting, or they found no choice. So what I want to point out is Grenova focuses on plastic consumables specifically. All you guys in here more likely have come across one of those consumables on top, and many more. If you think about how much plastic we put in the landfill, if you go ahead and multiply that by a number of samples you run on a daily basis, it's going to be quite significant. Especially when you consider all these different labs and across the nation as well as across the globe. Today I'm here to talk about the one tool that everybody uses, and that's the pipette tip. It's a tool that we all need in order to transfer samples, run our assays in some form, in some shape. And what's interesting about this pipette tip, it's been around for hundreds of years, and initially it used to be glass. We used to wash and reuse the glass and never throw it away. What happened? When the plastic came around, everyone started throwing it away. Was it because it was cheap? Or was it because the quality wasn't good? If the quality wasn't good, then it wouldn't make it into our industry. If the plastic has been used for making pipette tips, if it was a low quality plastic, it would not stand the chemicals that you guys are using inside the lab. We do question everything on our daily basis about the results and assays and everything else, and always trying to make incremental improvements, but we never question why are we throwing away plastic. And what I came to find out is plastic is a culture that has been embedded in us. Because it's plastic, we throw it away. How many of you guys have Nalgene bottles? How many of you guys throw away Nalgene bottles after one-time use? It's the same kind of plastic that the pipette tip is made out of. It's a polypropylene number 5. You don't throw away Nalgene bottles, but you throw away a pipette tip. So just give you an idea on how much pipette tips goes into a landfill. This is the trend for the past almost 10 years, and that's the forecast for the next couple of years. Right now, by the end of 2018, according to a recent report, there were over 77 million pounds of pipette tips. It's going to end up in a landfill. And it's on a steady rise. And this is based on the report that we've been able to gather from accredited labs. There's so many labs that we were not able to reach and find out how much tips are they using. So actually that number could be way more significant. But let's put it into perspective. 77 million pounds of pipette tips. These are hollow cylindrical plastics. They barely weigh anything, but take a lot of room. We did a math based on average geometry of the tip. We figured out how many tips they are. And we create a model, place them next to each other, and it will circle the earth over 10 times. That's how much waste we're putting in a landfill every year just from the pipette tips. And besides just the waste, it's a pretty big cost. Every time you run your samples, for every one of those samples, you use minimum 1 pipette tips, but usually you don't use 1 pipette tips, you use multiple. For every reagent you're adding, just throw everything away. And that's what Grenova was founded. We focus on the most common consumables that the labs are using, and that's the pipette tip. So we made our mission to not try to offer a service for you guys, to burn more energy, to come pick up things, recycle things for you guys. What we did, we said, like, well, what if, like, we give you devices, automated systems that you can safely wash and reuse in your own facility. You don't have to discard it. You just reuse it. But the challenge was the industry is like rapidly growing, number of samples increasing. So the products that we're going to offer, it has to be automated, it has to be high throughput, meet the demand, as well as reliable. And that's what Grenova was founded based on. It was a group of engineers. We came together and we focused on developing a fully high throughput automated tip washers that can clean and sterilize the pipette tips for reuse purpose. Our systems can do about 384 384 pipette tips within 10 minutes. That should be able to keep up with your throughput. But the other nice thing is you become independent. You don't have to buy tips anymore. You don't have to pay for shipping. You don't have to deal with oh, it's out of stock. You can sustain and maintain your own inventory in-house. You don't have to pay to discard it. You don't have to pay for to buy it. You bring it in and reuse it as many times as you want. But when it's time to discard it, instead of discarding into the biohazard bin, considered as a biohazard waste, and have to pay for it for somebody else to come pick it up and they do their cleaning or sterilization or however they're going to deal with it, you can just clean and wash those pipette tips one more time, because our systems not only washes but also sterilizes. They become a clean plastic. And then you can hand it over to the recycling centers. And that's something I'm working on, working with the recycling centers to be able to be open to start picking things up from the labs directly based on using those systems. Go ahead.
How much waste do you produce while washing the tips?
Excellent question. So we use roughly about 1.2 to 1.5 liter per rack of tips, but that doesn't consider to be liquid waste for two reasons. One is the cleaning solutions that it gets used during the wash process. It's a stabilizing buffer, so the pH that comes out of the back of the system is between 7 and 8. Also, all the liquid wastes are exposed to UV light, to a germicidal lamp to take care of any biological hazards or so. And this has been tested and verified by highly credited institutes such as CDC, NIH. Again, if you are dealing with compounds and you're worried about your compounds, you don't even want to take a risk of putting a small amount of compound down the drain. The system actually has two waste output on the back, has a primary and a secondary, and these automated tip washers are fully optimizable systems. You can program the wash protocol based on what your need is and how it's going to work for your sample types and for your tip types. So the most contamination from the tips, if it's a compound in this case, for the first 20 seconds that we are flushing the tips, we can collect it into a separate container for very small amount. The rest can go down the drain. So you're not introducing significant large liquid waste. So since 2015, we started implementing and introducing the products to the market. There are over 62 million pipette tips. It's been washed and reused among the labs. These are production labs. And this number is based on end of June of 2018. At the end of 2018, we're going to get a new report from our existing customers, and that number we're expecting it to reach about 100 million. We have helped the industry among all of our customers to reduce their pipette tip spending overall as an industry by $4.3 million a year. That's a lot of money that you can reallocate to buying instrumentations, creating more jobs, expanding on your assays or on your labs. But if you convert 62 million tips, it's equivalent to 170 pounds of plastics. Into perspective, it's over 6 million plastic bottles. I know every day, all of us here more likely, we pay attention if we're putting our water bottles in a recycling bin versus regular trash. Next time we look at the pipette tip, treat it the same way as you treat the plastic bottles. Currently, there are over 17,000 patient clinical samples are being run daily in the CAP and CLIA certified labs. These are all the different sample types and the assays. So far it's been validated and it's reported back to us. We are we are the manufacturer and engineers of these products. We don't have a lab, and it's our customers who are reporting back to us that like they're getting such a clean, good results by washing and reusing tips in their own facility for all these different sample types. And we just keep learning, like, what is the limit? The cell culture is one of the newest one. It's been added to our list. Again, we were not aware of it, but the customers implemented, they validated, and now we're going to add it to our list as well. But just like anything else, these devices, you validate it in your own facility. Like, for example, like if you buy a liquid handling system, or even purchase a manual pipetter, you don't just out the box start using it. You do verify, you validate, you make sure it's performing the same way as your standard. It's the same thing. So you validate the devices, these high throughput automated washers in your own facility, and compare the results for the wash tips versus versus the brand new tips, and you should see exactly same performance without any carryovers. But what if you do find better results with the wash tips? Who here has verified how clean the brand new tips are? We don't. We open up the packing and we just assume that it's clean. They are clean. They are manufactured in the clean rooms. They are done according to the standards. However, they still come from factory settings. We've had a couple of accounts that they implement our product to just wash the brand new tips to run their assay because they're getting less number of reruns. So it's a very interesting findings that we're discovering by just introducing this device, and we start now going back is did anyone ever verify how clean these plastic consumables in the first place? So the positive impact, the direct positive impact for your lab by implementing the Grenova products is that on average, you're going to get over a 90% savings, reductions in plastic waste as well as saving on spending. What we found is on average, labs have validated reusing tips 10 to 25 times. We have not had a case that they stopped the validation because either the pipette tip broke down, there was an issue with the carryover, they just got tired of validating. At 25 times, you're at 96% savings. At 10 times, you're at 90% savings. So most labs, they decide to at 10 times, they stop. That's pretty significant savings. So the financial impact for your organization is pretty significant. That's a bottom line net profit, about 3 to 4% increase for your company. And the plastic impact is roughly 16% of the plastic consumables in the lab consist of pipette tips. Again, this is an average. It could vary from lab to lab. So, but that's what we found based on the industry average. And what we've done since the introduction of these products, we constantly getting requests from our customers that can you wash these tips? Can you wash this brand? And what we found is all the tips are actually very similar. So in some cases, I wonder if they all come from one or two tip manufacturers, and they just get repackaged. But these are all the different tip types that so far has been validated, and we are aware of it that our systems are in production and they're validated for reusing these pipette tips. All the way from manual pipette tips to automation tips. One thing I want to point out is by implementing our product, nobody has to wear gloves and start picking up pipette tips from a trash bag and start racking or so. The tips are always washed in the rack. So for the automations, for liquid handling systems, all of those robots are programmable to be able to eject the tips either back into the pickup location or to an empty location, empty tip rack. And all it is, all 96 or 384 goes in there, you just pick up that rack and place it inside the system. So there's not much of a big effort anyone's has to dig into the biohazard bin. For the manual pipette tips, it's just a change of hand movements. Instead of when you pipette, you go like this, now you just have an empty rack on the side and you just eject it back to it and pick it up and place it inside the system. Quickly give you an overview of these products. These are benchtop systems, very compact benchtop systems. The largest system is about 2.5 feet by 2.5 feet. And it can wash anywhere between 16 to 24 tip racks. Again, because the range is based on the wash protocol that gets optimized for you based on your assay. TipNovus Mini is a system you can actually integrate it with your liquid handling systems with the gripper arms to make it fully walk away setup. A couple of images from our existing customers. This image on the bottom. It's very interesting. This lab, what they did. So The workflow, that's one of the things, that's one of the interesting parts of we've seen like how labs implement these products as far as being sustainable and reducing cost, how they implement it within their workflow. Some they put it within their production facility or right next to a liquid handler or like, at the end of the station where they do the pipetting or so, and the techs handle the pipette tips. And some, what they do is like they create a wash facility. Literally, they create a washroom. They put a room together, the eye water source, and put a couple of systems based on their throughput that they need. And they have tip washer positions in the company. And they save significant amount of money. Like this lab, specifically with 4 systems, are saving over $1.1 million a year on pipette tips alone. But what's nice is they've created a job. They have 2 shifts. 2 individuals working like 2 shifts and managing all the pipette tips within a lab. So the scientists, during the day, they don't even have to deal with handling the tips or washing the tips. The tips gets managed by the tip washer manager. Just comes in, gets picked up, brought into the washroom. They get cleaned, they get stored, and then they create a backlog of a couple of days of tips, and they just recycle it back into the lab. Again, they were first verified that the cleaning and sterilization is done by our system is 100% reliable and it's validated. Then they implement the process. So that's all I have for today. But let's just look at a new way, new approach. What I like to call is a smarter, cleaner, and a greener approach. So everyone ready for a tip washer? Bring in. We're going to have a table back there. My colleague actually, she just arrived, so we're going to set up a table, and we have brochures, we have results, we have data that we can share, and look forward to working with you guys.